Hello and welcome to the Science Club show. We are here today with Ben Kiernan, who for some reason is just not showing up on the good old stream here. There he is. Hello.
Hello.
You are now on the screen with all of the people watching. So Ben, you were with us. Yeah, it's always good to have like fellow podcasters and stuff. You were here, of course, on the last show, the first show that we did, and now you're sort of back again. And it's been a very interesting time since then. A lot of predictions got made on that show, and now we're kind of seeing the course of the coronavirus, of course, going through. And we actually did a podcast that is out now. On the non-peer review. Yeah, and you had myself and a medical doctor on there to talk about coronavirus, sort of generally, trying to dispel some myths and get some good information out there. And that's about an hour long. And I think it's a good resource as well. And obviously we've talked about it on the Science Club show. But no coronavirus talk this week, directly at least. We want to give people a bit of a break if we can. But we do have a lot of stories. But I guess the thing that is affecting us, even without directly talking about coronavirus, is the sort of social isolation and working from home. You work in science media and reporting and stuff like that. So I'm guessing that you have been working from home as much as possible in the past few days. How's that been going for you?
Yeah, so we transitioned fully to working from our own homes on Monday. Half the office had been working from home before that. And the transition's been interesting. We've been having a lot more video conference meetings than previously. We had this morning, we had coffee together at in video conference format, just in our own rooms, drinking coffee together.
Yeah.
It's working where one of the things that we do at my job is we do these media training courses where we connect scientists and journalists and get them to talk to each other in the same way. Because scientists don't know how journalists work, and most journalists don't really know how scientists work, and they also work on completely different schedules and time frames.
Yeah, of course.
Because scientists want everything in writing and then have enough time to review, and journalists want it today because it has to come out tomorrow. And so we get scientists to hear what the journalists, their point of view, their perspectives, but we can't run those courses with the isolation going on. And so we're looking at a kind of online presentation type format. So that'll be interesting to see roll out.
It's kind of interesting, yeah, the sort of this shift to online. I mean, one thing that I've definitely discovered quickly is that whoever designed those kind of conference rooms where they have the Zoom setup clearly has no idea about acoustic engineering and sound dampening and all that stuff, because it's all just this big echoey mess. So I feel like a bit more science could have gone into those rooms, but also kind of the way it's changed, I think, the way we look at interpersonal communications has been interesting. And yeah, it'll be. I guess as time goes on, it will be interesting to see if any sort of science and psychology comes out of this. Because we're all doing something en masse. We have these huge amounts of data points now, and I imagine there'll be a lot of studies going on, gauging people's psychological progress as we have to sort of stay in more and more. So
yeah, it's going to be interesting to see once we are no longer forced to be isolated, how many businesses are going to keep a similar situation, because obviously hiring a room and maintaining office space costs money.
Yeah.
And if you can have part of your workforce working from home and they're happy to do so, then how is that going to change workplaces in the future?
Yeah, exactly. And I think a lot of industries probably will move to more home-based kind of work. I think once we realize it can be done, obviously some of us can't. Like I can't work from home a lot of the time because I'm physically in the lab. And, you know, I can't say too many particulars, but, you know, I'm working with a group of people who are important to the government response to COVID at the moment. I'm not directly on the project myself for now, but maybe if things get too hectic, who knows what could happen. But yeah, we're sort of working hard at the moment, trying to make sure we can test as many people as possible. So yeah, it's been an interesting time. Yeah, hello from Canada, Echo. So I'm going to say Echo Dubleg, or I'm not quite sure how you pronounce that, but welcome to the show. Very nice to have you. Please, if you're in the chat, feel free to comment and ask questions throughout the show. And if you have an interesting science story that you've come across that you'd like us to look at, we're more than happy to sort of do it live, as it were. That's kind of the nice thing about the show compared to the dailies that we have on YouTube. We get to sort of interact, and it's really nice. So the first study I actually wanted to talk about is, it's a dark topic, but it's good news about the dark topic, if that makes sense. So Ben, I think this is kind of something that is definitely culturally in a lot of people's, at least the sort of the cultural knowledge base. But are you aware during the Vietnam War that a lot of landmines were buried essentially in Cambodia and Vietnam and the surrounds? And it's actually,
and not just Vietnam as well, other wars too.
Yeah, absolutely. Cambodia is a famous one, but other surrounding countries as well. This is a big issue, and you know there are a lot of landmines still, and in essentially the 60 years since the bombings in Cambodia and Vietnam, 64,000 people have been killed or injured by unexploded bombs that are essentially still buried in the ground. And it can be hard because obviously these were designed to explode when people step on them and people are working in the fields and things like that. Still, and you know, while those zones are demilitarized now, you know, for a long, these mines are still really doing a lot of damage, and it's a really expensive process, of course, to try and find these mines and identify them and try and remove them, because you need to do that. I mean, it's kind of an obligation, really, isn't it? You know, the people involved in the war have this obligation now to try and make the world a better place since then. And so really finding ways to actually get rid of these bombs is really important. It turns out that if you look at some declassified data from the US military, there are some 44 to 50% of bombs in the area that actually are unexploded potentially. And the cool thing is that what I wanted to talk about here today is that there's this new method, and it's actually using satellite data to identify these sort of bomb craters, you know, these little indentations where the bombs have been buried, or just the bombs that were dropped, you know, not just mines, but bombs themselves that were sort of dropped and they're now in the soil. And it's actually really cool because it uses a technology that I'm personally a big fan of, and that's machine learning. And essentially what they've been able to do is they have been able to actually use machine learning to sort of learn what a crater looks like that actually has a bomb in it. And the cool thing is that this method currently has 160% better detection rate than the standard methods that we're using now. And obviously the methods we're using now aren't especially effective. I mean, I'm sure they're quite laborious and careful, but this technology...
I can think of one of them which isn't so laborious, but it's more biological.
Yes, not really the one you want to use.
Yeah, well, no, not that one.
Ah, okay.
There is a plant that was engineered to, when it is in soil that has, I think it's a higher level of nitrogen or something that is found in these explosives, then its foliage is different. It becomes, I think, red or a different color to what it would normally be.
That's amazing,
but you have to wait for it to grow across a field, and there are lots of factors that maybe it's just a bad year and it doesn't get as much rain, and so like you get these false positives. And like maybe it just passed over an area that has high levels of nitrogen or whatever it is it's looking for.
Yeah, that's actually really interesting, isn't it? Like the way that plants take in these different nutrients and based on what can be quite small changes, right? I imagine that the difference between where the bombs are and where the bombs aren't is quite small in terms of like soil terms, but the plants are sensitive enough that they can actually do this kind of thing. And so I think it's kind of one of the interesting things is that the researchers actually used these algorithms originally to detect meteor craters on moons and planets. And essentially they managed to finesse this model to kind of move it to the bombs, because the bombs create similar looking craters to meteors, just in a smaller form. And it's kind of really cool how the discipline of like looking for things in space actually has kind of helped us with looking for things like bombs. And what basically what they had to do is train the model from looking at craters on the moon are obviously quite uniform because there's not a lot of things happening in space. So the impact is kind of the only thing that's happened on that surface, potentially for millennia in that one particular spot, especially one big enough to be detectable from the kind of ranges that we can look at them, right? But obviously what happens is to these bomb craters is that over time, as you say, grasses and shrubs and things have kind of grown over them, and rain has created erosion. And that was kind of the key step in training this computer algorithm. And what they were able to do is use that to kind of build on what they'd done with the moon craters and the meteor craters and things like that, and sort of get the machine to sort of learn them. And typically how these things work is they use something called an adversarial network, where there are like two competing algorithms, one that tries to like guess what the data looks like and one that kind of knows how accurate it is, and they're both kind of fighting against each other to try and sort of constantly sort of, you know, like trick one into thinking what they have is real, so that the one that is detecting gets really good at detecting the real thing compared to the one that isn't quite right, essentially. Really cool stuff. And obviously image recognition is only one way that AI is being used. But other ways are obviously in things like recognizing language and voices. They're used in science a lot to try and like see in a sea of information, you know, when you have so many genes that might be associated with a single thing, they can identify the particular ones that are actually responsible, say, for a specific disease, because it can sort of detect these patterns over time. So
I think they've used similar technology to look for ruins in jungles and rainforests, because they're able to see the patterns that satellites can pick up you can't see from the ground.
Yeah, and I think it's the satellite data, the fact that satellites have such good imagery now is helping along this process massively, which I think is really great. But hopefully this results in a lot of bombs being detected and you know, decommissioned, you know, so that we can sort of save lives and people getting injured. So it'd be good to see if these things actually end up working. So the next story that I have here is kind of an interesting one. And it's actually, when you think about it, it makes a lot of sense. And I think the important thing is with this research actually is what we do with the research rather than the research itself. But the sort of headline of the research is that chattier kids do better at school, which kind of makes sense if you think about it. But there's some interesting like socioeconomic reasons why that is. So essentially, what the research looked at is the verbal skills of young children going into school, and they tracked them all the way to the age of 16 to see how their academic career progressed. And this was done by researchers at the University of York. And essentially what they wanted to know was why children from wealthier and more well-educated family backgrounds do better at school. And some of these kind of make a lot more sense, you know, like intuitively. If you come from a well-educated family, there's a good chance that that family is going to bring you up with a sort of sense of appreciation for education, right? And that probably will help you in school. and wealthier families have more resources to put into to their kids. And essentially what they found is that the children from families of higher socioeconomic status, they actually had these better language abilities before they even came to school, basically. By the time they were school age, they already had the sort of better verbal skills than people with the lower socioeconomic backgrounds, which obviously translated later to better academic performance. And I think and I think one of the things is as well is that obviously teachers are well-educated people, you know, having gone to university and that kind of thing. And because of that, there's a certain kind of language that teachers are going to use and kind of in another way kind of expect back from kids. And so kids that have these backgrounds to begin with, they're more likely to already have this vocabulary, I guess, and this connection with the teacher in order to sort of get the most out of the teaching as well. And they actually found that these differences in language skills actually explained about 50% of the family background in terms of children's achievement, which I think is a huge actually amount. You know, when you think of a lot of other things that can happen, you know, distractions at home, lack of resources, or like, you know, lack of ability to do things like extracurricular activities, because obviously they're not cheap, and a lot of families can't afford to do those kinds of things with their kids because they just don't have the resources. And obviously kids that are better off, they tend to do more extracurricular activities and can therefore do pretty well. And
so my question is, what do they define as chatty?
Yeah, so I think chatty might be a sort of editorial choice of the publicity around the study, but basically kids that have better vocal skills, I guess. But also I think if you think of the idea of what chatty means, I think it's basically not too controversial to think that kids that are a bit more sociable at school, especially when they're young, I think they tend to get more attention from the teacher as well, right? Because kids that are more likely to keep to themselves, that I think they're less likely to draw the attention of the teacher. And so perhaps kids that are a bit more endearing because they're a bit chattier, they maybe get that extra attention that you might not get if you're a bit more reserved. Because obviously there's a lot of competition in a classroom, right? Like we have classrooms that are getting to 30 plus students in a class, and that's a lot of individuals for one teacher to try and focus their attention on. And no matter how hard a teacher tries to sort of be equitable, I'm sure it's not easy to give every single child the same amount of attention. And if one particular child is just a bit more amiable and chatty, and also, you know, kind of has that rapport with you because of their background, I'm sure that is going to also lead to just slightly different amounts of input from teachers to these kids.
Yeah, that's true. That makes sense. I wonder how like introverted, extroverted, chatty, like, because I think this is without any kind of information to go on, but it seems that introversion starts kind of later in development. And so, like, because introversion isn't necessarily people not wanting to talk, it's just not wanting to talk in certain situations. And so, like, if a teacher or the researchers got an introverted student into a situation they were comfortable to talk, would they become chatty in that situation? Which I think, yeah, it's like chatty is across all spectrums. Because you can have very chatty introverted people, they just are introverted until they're ready to talk.
Yeah, exactly. I mean, I think that actually does have a lot of impact, because the classroom environment isn't necessarily the best for everyone. You know, it's a very intense kind of environment for a lot of people. You know, 30 people in a room. I mean, these days, I don't particularly love that situation myself. I mean, I was lucky at school that I guess I you know, kind of thrived in that situation, luckily. But as I've gotten older and my sort of, you know, tolerance for people has decreased a bit, I think, you know, that I struggle with that as well. And I imagine a lot of kids would as well. One of the interesting things I kind of found with this research is they also looked at some non-verbal skills as well. So beyond language skills, things like puzzle solving, being able to draw shapes, copy actions, sort of recognize things like body language. They found that actually these only accounted for a third of the differences between, you know, the family background and educational achievement, which is kind of interesting because it obviously means that, you know, your innate ability, I guess, to sort of be able to solve puzzles and things like this, maybe unlike the verbal skills, which I think are very social, that maybe these non-verbal skills are a little bit more innate than they are learnt. You know, things like puzzle solving and drawing shapes, like there may be a lot more effective natural aptitude on that than things like the vocal ability, which I think is one of the interesting things that this study kind of brings up as well.
Well, probably people from higher socioeconomic backgrounds have had more access to puzzles and more complex thought environments, I guess I want to say.
Well, yeah, imagine, you know, these days kids get access to things like iPads from a very young age, but if you are from a lesser socioeconomic background, you may not have access to those kind of games and videos that are aimed at kids to sort of kickstart the problem solving from a young age. And even maybe looking further back before the iGen, if you like, the kind of toys that you can get, you know, like the more complicated, expensive toys do tend to be better for stimulating learning and problem solving compared to cheaper toys that maybe don't have the same educational content that you might have access to. So I think there's a lot of reasons why you really are quite disadvantaged if you're not from a sort of well-educated or high socioeconomic family. And I think it means that we have to work really hard to put the resources into kids so that they don't fall behind and they have the same opportunities as much as possible. Because obviously it benefits everyone, and I think everyone deserves the chance to get the best education they can, regardless of their circumstances.
So, Ross, and I'm going to play with the frame here because I can see myself. I've got the stream running. Ross, were you a chatty kid?
I have to look this way. Yes, I was. I was a very chatty kid, Ben. I started talking fairly early, I guess, and I was quite chatty, and I would talk a lot I would actually have conversations with the teachers specifically a lot as well, which I think does help. And I was, as I said, I was lucky as a kid to be confident enough to do that, whereas, you know, a lot of kids naturally don't have that confidence, and even being from sometimes like a lower socioeconomic background can affect things like confidence to talk to people like that. I think, you know, like
that's true.
Seeing teachers a lot, I think you're more likely to see teachers as an authority figure who you have more reverence for, rather than sort of you know, like you have a relationship, whereas teachers, they don't kind of want to look down on kids. I think a lot of great teachers actually see kids as equals, and sort of the relationship, obviously you have to be careful that you're not friends with the kids, but you do want to make sure that they're not just, you know, they're not just merely pupils, you know, they're kind of humans that you interact with. And I'm sure a lot of teachers would agree. And Tina, who is unfortunately stuck in the lab today doing late night science, is laughing at our little play of looking at each other through the internet. The funny thing is, when I actually look over here, I can't see you at all anymore because the screen is in front of me, which is quite funny.
So what you need is a screen. Hang on, it'll be over there for you.
Yeah.
So that you can see what I can see when I look this way, and I happen to have, out of the peripheral vision, I can see my glass door. So I can kind of see this. Yeah,
just this sort of weird virtual, or maybe the future of these meetings is like the VR headsets. So that would be interesting. So yeah, I think that would be actually quite fun in the future, because like the 3D, I think the 3D effect of like actually kind of looking like you're sat in a chair watching would be really cool.
Well, because what would be great would be if, like, you and I, Ross, if we had full motion capture and people could get into like a lecture style and we can stand up in front and walk around and point to people.
Well, I mean, with the current social isolation situation, maybe that's how universities are gonna go from now on. Because I definitely have seen. I'm sure you're still on a lot of university groups, Ben, yourself. Students are not loving Zoom lectures.
I think lecturers are not loving Zoom lectures.
Yeah, that's true as well, I guess. Like a lot of lecturers have had to learn this new technology very quickly. And yeah, it'll be interesting because there's definitely a lot of research while we're talking about education that shows physically turning up to a lecture increases your probability of success, in exams and things like that by quite a large amount. I forget the exact percentage, but it's a fair bit compared to people who only watch the videos. So yes, I would be interested.
One of my good friends was telling me about, because he's currently going to university, he's in a group Slack now, I think, like for his subject.
Yeah.
And somebody in the general chat commented something along the lines of, so what program are we actually using for this subject? And that was 2 weeks into the subject and the day of an assessment due.
Right. Yeah. I mean, that's just kind of student life in a nutshell, though, isn't it? I think because you tend to see that a lot. I mean, I've done like tutoring in a lot of subjects, like undergrad tutoring and working in prac classes for microbiology and stuff like that. And it's amazing the number of students who just really don't switch on until right near the deadline. And admittedly, I was one of those students as well back then. I feel their pain, but at the same time, knowing how I used to approach university, I always tried to sort of discourage the students from approaching it the same way. But obviously, they're adults.
Every Zoom lecture starts with 15 minutes of "Can you see/hear me?"
Yes, which is extremely true, and it's really awkward. We're lucky that we get to do that offline, as it were, on the show.
Yes.
While you guys are watching the last week of Science Dailies, which you can find on YouTube, Science Club Network, we are in the background trying to make sure OBS looks fine. And for some reason today, even after we did all of that and we went live, it still didn't work properly. We still had 30 seconds of me like making sure Ben wasn't invisible. So yeah, the technology is interesting, that's for sure. Well, let's talk about something not technological, the opposite in fact. And I really want to talk about this new fossil that has been found. I believe it was South Australia. Yeah, South Australia. And this is really cool to me because this is one of the earliest fossils we've ever found, about 550 million years old. And it's the first bilateral animal, basically. And I'm sure to a lot of people, none of that means anything. So I guess I'll kind of explain a little bit.
As a physicist, I understood the word animal.
Yeah, or maybe bilateral, right? There's like bilateral symmetry in some things, but not in this case. So yeah,
I know those words, but just not together.
Yeah, not in this particular sentence. So essentially, the way animals evolved on the Earth, at least, we don't know about anywhere else as of yet, is that we started off, of course, as we think basically little pieces of DNA or RNA just sort of surrounded by fat. And then from there, proteins kind of came in and sort of helped the replication along. And then most life for the longest period of time on Earth, like over 2 billion years, was single cell life. Just little cells that were all by themselves. Some of them utilized light, some of them utilized eventually oxygen and various nutrients, but life was unicellular, just single cells for the most part. And then somewhere around sort of 500 to 700 million years ago, not billion years ago, the universe isn't that old yet, about 700 million years ago, we started seeing the formation of multicelled organisms that are the precursors of animals as we think of them today. And one of the important innovations in these sort of colonies of animals, as it were, and to be fair, the bilateral animals are actually only still one branch today. Like there's the derriere and the sort of some of the more weird sea animals that still don't belong to this group. But essentially what happened is a certain group of these animals, about 550 million years ago, they formed into this bilateral shape. And what that means is that the cells organized themselves in essentially these two identical halves. And actually the best way to think about it, like literally just off the top of my head just now, is that they're kind of like a Zorb ball. If you imagine that shape, that's exactly what the most basic animals looked like right at the start. So essentially these spheres with like a cylinder going all the way through. And if you can imagine that exact shape with like the sort of invagination at the top and the bottom of the Zorb ball, that's almost exactly what
like a dumbbell, like a weight.
Yeah, essentially. But like obviously with like a hollow tube, and the hollow tube is the origin of the mouth and the anus. Like they were actually some of the first animal structures to develop. And I guess the benefit of this, evolutionary speaking, was getting clean nutrients from one end and depositing waste on the other meant that you produced a lot less toxicity within the colony of cells than colonies that were contained and essentially had to have less efficient ways of expelling waste. But one of the things that's really important in biology is this idea of gradients. And so by creating gradients, you had a way of getting rid of the waste, and therefore you may be better able to survive. And the cool thing is that this fossil, and I know when people think of fossils, they think of like dinosaur bones or like, you know, those little kind of bugs that you can basically buy because of the Cambrian explosion, those little
Armanites.
Yeah, exactly those things. They're so ubiquitous that pretty much anyone who wants one can just buy one, which I think is really cool. I think everyone should own one of those. Like fossils are the best. But this thing is actually literally just the size of a grain of rice, between like 5 to 7 nanometers in size. And one of the interesting things about this is that even at that small stage, so this would be a very early version of this, it had kind of a head and like a backside, as it were, and the head is kind of a little smaller than the backside, which is sort of larger at the bottom. So if we go back to that dumbbell example, imagine like a smaller piece, like a smaller weight on the top, the tube, and then a slightly larger bit at the bottom. And obviously that mirrors eventually how a lot of animals have evolved over time. And it means that this particular shape is clearly very evolutionarily conserved because it has a lot of use, I guess. And I guess because in a lot of ways, you know, you want to sort of restrict the nutrients coming in, but you don't want to restrict the waste coming out. You know, getting rid of waste is a really important process. And what I like about this is that they didn't just find the actual creatures, but they actually found these like little burrows. So if you imagine like worms, these worms are kind of like, in a lot of ways, haven't evolved much past this sort of initial like bilateral stage. Like a lot of worms are still essentially this very basic configuration, but it obviously means that configuration is very successful because worms are so ubiquitous. But I love the fact that we not only found the fossils, but like these little evidence of the lifestyle that they had, you know, kind of burrowing through. And obviously, like pretty much everything at that point, they were soft bodies because they hadn't evolved things like chitin or like keratin or anything that
skeletons or anything like that.
Yeah, exactly. You know, like a lot of life still, of course, has no hard structures at all. Like we talked about slime molds on yesterday's Science Daily. You know, they don't have any like hard bits. Most of life on Earth doesn't, in fact, but obviously these things being very early didn't. So yeah, I think it's kind of interesting that previously all we had was the sort of the burrows that they made, and we could infer that they existed, but we didn't actually know. But for the first time, and it turns out this particular place is really rich in fossils. So it's like a really good sort of treasure trove of these fossils. But it's kind of interesting that for the first time we sort of see like the head and tail, as it were, of an animal. And I think that it's, I just love kind of seeing these animals, because to me, evolution is one of the most fascinating topics in biology, because it is essentially the driving force of biology. Like the thing that makes biology exist is the fact that at some point in Earth's history, DNA existed, and because that DNA or RNA got the ability to copy itself, the second that happened, evolution had to occur, because the DNA or RNA that could copy itself the best would keep copying itself and compete better for resources with the DNA and RNA that didn't copy itself as much. And that meant at the start, probably the actual DNA and RNA, their own structure is what made them copy themselves. And then over time, they got maybe picked up these protecting like lipid envelopes, or the proteins came in and like made, like basically things that just happened to be enzymes got kind of captured, and that helped as well. And it's this fascinating chain that goes back, like 4 billion years, basically, maybe a bit less, maybe like 3.7, because the Earth was quite, like a lot of turmoil on the Earth back then. And, over time, these things just sort of, because of competition for resources and the need to copy itself, just keep changing in all these different ways. And I just find that sort of chain, the unbroken chain, as it were. I think Carl Sagan, as is often the case, just, really put it beautifully in the, that the unbroken line between that first ancestor and us that's just continued for over 1 billion years is absolutely amazing.
Definitely. Part of my science communication brain, just the back part of my brain, has been kind of spinning wheels since you mentioned it. And so now I have to get this out of my brain. So what you're saying is you like big biological gradients and you cannot lie?
I would definitely say that, yeah. That is definitely true. Yes, very well done. Very well done.
Thank you.
So what
I was going to go with another, like maybe a little bit lower, something about the worm being thick.
Yeah, I mean,
it's dumbbell thick.
Yeah, I liked the biological gradients. I thought that was pretty good.
Thank you. That was the higher level one that I went with.
That's definitely like the next level stuff. So I think the one thing I just want to mention, which I think is pretty cool, just before we leave off this topic, is that in the burrows they actually can sort of tell by looking, I guess, at the molecular structure of the burrows, is that these animals were actually not just moving randomly, but they actually were moving specifically towards food and oxygen. So even these like really basic rice-sized animals had the ability to sense the environment and move towards things like food and oxygen, which makes a lot of sense because even single-celled organisms can sort of see and smell.
Well, this would be a multi-cell organism.
Of course, yeah. But what's interesting is that even some bacteria can sense light or can essentially smell. They can detect chemical gradients, which is all smell really is. And so, yeah, you can. It's kind of great how even really early on that these animals had the ability to sort of have this kind of self-determination long before this like advanced nervous system would develop.
I mean, we still see animals that lack what we would define as a nervous system doing complex things. Jellyfish don't have a brain.
Oh, absolutely, yeah. Like the way cells communicate and as an organism can function as one without, something as complex as a brain, like really basic nerve-like cells is actually...
I mean, even plants react chemically when they're attacked.
Yeah, like grass. Like the smell of cooked grass is actually kind of a war cry, isn't it? Basically,
petrichor.
Yeah, which I think is just like fascinating by itself. And yeah, we obviously we had Debbie on the last Science Club show who talked a lot about the various things that plants can sense and do, which is really cool. I think plants are very underrated actually as in biology. I remember groaning when we had to learn plant physiology, but now I'm a bit more into like science in general. Plants are actually really cool. Well, let's talk about still some small animals, but these are not animals. In fact, they're bacteria, and this is kind of an interesting result in that the thing itself is kind of like normal medical stuff, But I think what's cool about it is kind of the implication it has for the way we look at proteins and like what their role might be. So essentially what researchers have found is that using a model in mice, they've been able to see that a fragment of a protein, not even the entire protein, but a fragment of a protein that is created by bacteria can actually lead to pulmonary fibrosis when the mice are infected with this bacteria. So pulmonary fibrosis is kind of this, is a medical condition that is sometimes associated with smoking and asbestos, but in a lot of cases, actually, it turns out a large number of cases, they actually don't know what the specific cause is. But it's actually not, it's a really sort of bad disease to have, because it turns out that 50,000 patients in the US die of this every year, and the survival time in 5 years is really poor as well if you have pulmonary fibrosis. And essentially what happens is over time the lungs get sort of attacked, and instead of being made of this sort of spongy light tissue, which the lungs are made of, which obviously is really good for bringing in air, you know, that sort of surface area thing, the surfaces start becoming harder and more fibrous. Like imagine, you know, what happens when you scratch yourself and you get like a scar. That kind of tissue can form in the lungs. And of course, the more that happens, the less oxygen that you can get. And it essentially turns into this thing called acute exacerbation, which is, I guess, similar also as what is happening to some of these coronavirus patients. And at that point, after a certain amount of time, unfortunately, when too much lung tissue dies, you can't get enough oxygen, and obviously the patient dies, which is something that you would want to avoid. And what they found is that in the past, some people with this pulmonary fibrosis have actually had particular bacteria that live in the lungs. Especially, it turns out, salt-loving bacteria or halophiles. And that makes sense because the lungs actually are a surprisingly salty place. There's a lot of chloride and sodium in the lungs, especially in the secretions that the lungs create. And a lot of those secretions are actually to keep bacteria from getting too close to the actual membranes of the lungs itself. And essentially what they found is that these bacteria all secrete this particular protein, but the protein itself doesn't actually lead to the fibrosis happening in the mice. So they looked at basically these bacteria called Halomonas, Staphylococcus and Streptococcus, and these are particular ones that are a bit more like I say, halophilic, which means like salt or salt-loving. And what they found was when they infected the mice with the bacteria, they would develop the pulmonary fibrosis. And what they found was that there was a particular like peptide, they call it. So things that are smaller than proteins are peptides, and they called this peptide curacin. And these bacteria produce this, but they only actually cause the disease if the bacteria were present. That was one group. Obviously, they had a control group. If the whole protein that this coursing was derived from was present, the damage didn't occur. And so only if the bacteria was there, which means the bacteria produced the protein and then clearly they cleave this little bit of the protein off to create this curacin, or if the curacin itself, if the researchers produced the curacin and infected the mice essentially, applied this to the mice lungs, then they found that this peptide actually ended up creating this fibrosis. And so what they think is happening is that this peptide curacin actually interacts with the cells in some way and triggers the lung tissue to undergo various sort of self-damage that then, like a process called necrosis, and then the necrosis, then the body's response to try and protect from that is the fibrosis. But because the bacteria are kind of still there, or this peptide fragment is still there, they actually, this process just keeps happening until unfortunately the fibrosis goes away. So I think the interesting thing for this though is that obviously, like finding out what this is means that they might be able to find a way to neutralize this peptide and find if the people who have fibrosis early have these bacteria. Maybe they can try using certain drugs or chemicals or something to try and like curb this process. But I think one of the interesting things is that when they were trying to look for the way this protein functions, is that if you were to actually look at the genome of this bacteria and look at the whole protein structure, you would actually not be able to find this peptide, because peptides get cut after the protein is made, and it's hard without just looking at the gene or the actual protein sequence itself to actually find this stuff. And so I think that means that when we're trying to find the causes of things like this, people studying these actually have to look a little closer, and maybe actually when they're looking at samples, they have to do things like mass spectrometry, which is a way of essentially firing molecules at a screen, seeing how long that takes, and discovering what the molecules are made of. And in that way, you might be able to find more things and do things like gels. So for me, it was really interesting that if they hadn't sort of done an approach that allowed them to detect this peptide specifically, and all they did was look at genomes and whole proteins and stuff, they never would have figured this out. And of course, you know, the study itself of separating the protein from the actual peptide is really good scientific work. Like, it's a really nice control to sort of prove the fact that the coursing is the thing that is doing the damage.
Mm. Okay.
So, sorry, I'm just ordering my dinner.
Okay, so here is a fun science communication challenge for you, Ross.
Yes.
Could you explain that story like you were explaining to a grade 7, like a 12, 13-year-old?
Yes, so I realised that I used a lot of words in this story because it's kind of complicated. So I guess if I was kind of say it to a 12-year-old, and I'm going to give 12-year-olds a bit of credit, is that essentially there is a disease that can happen in the lungs, which is what you use to breathe. And what happens is over time is that unfortunately the lungs get damaged, and when the lungs get too damaged, the lungs don't work well enough anymore and people die. And what happens is that, you know, bacteria, which are sort of little cells that exist everywhere, can sometimes get into the lungs, and they produce this little structure, and this structure is called a protein, and proteins are what all of life is made of. And sometimes these proteins can be cut into smaller chunks. And what these scientists found is that the smaller chunk, but not the whole protein, is what is causing damage to the lungs and ends up in this disease. I think that would work. I think that's probably enough. It's probably enough.
I think that's good.
Yeah. So yeah, hopefully that would do it. But I know that that's the thing. It's really hard when you're talking about these really specific medical cases, because I think that's the thing with science as well, right? That as you get further down the rabbit hole, the words get so much more specific, and you have to sort of explain more and more about a story when you get into them, because the story itself doesn't just make sense to the average person. It you know, like most people wouldn't really know what a peptide is, you know, just on everyday life. I think a lot of people might know what a protein is, but even I think the concept that life is made of proteins, and that proteins form bigger structures, and those structures form cells, and cells form the body. I actually don't know that if you were to do one of those, you know, like segments where you asked random people on the street, I don't know how many people would actually know even that part, you know, that is how life is actually made. You know, you start with DNA, and DNA leads to proteins, and proteins lead to bigger structures, and those structures lead to cells, cells lead to organs, and organs lead to bodies. Like, obviously that's a very basic biological concept, but I wonder how many people would actually know that is what life is, you know?
I think people would go from DNA to cells, and they would miss the protein in the middle.
Yeah. And it's funny because proteins are they're not easy to sort of talk about because they are actually very complex, which is interesting. You know, it's almost funny that proteins are almost the most complex part, and the way the proteins come together is almost easier from a biological point of view to explain. And as you get down to the protein level, things get very complicated because proteins actually aren't just made of those 20 amino acids. That's a lie, because sometimes there are weird amino acids in very particular cases, but those amino acids can change a bit. You know, you can actually, like, the body does these little things to the amino acids to actually change like whole shapes of proteins sometimes. And so, yeah, I think getting those concepts across can be quite difficult.
Ross, here's a question, because you know more about this than I do. I'm sure in your time you've seen plenty of animations of proteins like walking and moving. Why do you think, as an artistic choice, proteins look fuzzy?
Oh, you mean in animations and things like that?
Yeah.
Yeah, it's kind of interesting, isn't it? Because it's the problem is that it's really hard to visualize things at the molecular level, because things have a structure on the macro level. Like, you know, I can sort of hold up this mouse here. It's big enough that there's a really defined shape. But the problem is that, and I think you would know this from a physics point of view, molecules themselves, they don't have a shape so much as they have forces. And the way that things like, say, hydrophobic and hydrophilic fields, or, you know, covalent forces come together, that's what causes the shape of a molecule, as it were, because you can look at the individual atoms and they give you what looks like a shape. But then as soon as you add all of the forces that make up the sort of actual interactive part, that adds this whole other layer of, like, I mean, I wonder if I can actually try and find the sort of the different stages of a protein shape to get up on screen. But it starts off as molecules, and, you know, like those, I think you all have seen with those wire and ball things as a kid, right? The sort of, yeah, so I can actually try and get this up here. So the chain here, I'm going to have to, unfortunately, Ben, sort of cut you off from the screen for just a second. But
sure. I understand.
If we go to this, I can put this on the screen. This is the kind of typical wire and ball approach to looking at molecules. So each sort of atom is represented by a ball. So that's like carbons, hydrogens, that kind of thing. And so this gives you a kind of idea of the shapes. But in reality, we then have this next stage, which is this sort of where we represent the different types of structures. So proteins aren't just long chains; they form into these like helixes, you know, like little like coils. They form into these kind of sheets, and then they just have kind of the more linear structures, right? And then you have this sort of one where you actually model all of the interaction, and that's the one that's on the screen now in a lot of ways. And they're the really abstract ones where you're not just modeling the balls, but actually the amount of space in terms of force that they occupy. So yeah, proteins are very hard to visualize because they don't, like, shapes don't exist really at that level, interestingly. Like they exist in a very sort of force way, right? Where the way proteins interact is that, you know, one part of a protein may have an affinity for positive charge, and the other side has an affinity for negative charge. And as they come together, the forces kind of bend where all the atoms are. So it's a very
so I don't know specifically how big these are, but are they smaller than kind of the like wavelengths of light? So we couldn't use microscopes to see. You'd have to use something.
Absolutely. So
right.
proteins are, like, individual proteins are so small that you can't even actually see them very well with a scanning electron microscope. So, like, a virus is basically almost the smallest thing we can see a definite shape of on those. I think everyone would have seen those scanning electron pictures. And again, I can bring one of those up. And you can sort of see the sort of, yeah, these are really nice. Diatoms look amazing on scanning electron, and they're the kind of shapes that we can get, and like these sort of cell level things. But proteins themselves can't be visualized that way. And the way that we actually know what proteins look like is we turn them into crystals, which is incredibly difficult because proteins don't like to become crystals. And what we do is we fire electrons at them, and by looking at the way the electrons scatter, we can place each individual atom. And essentially from there, we can figure out which atoms are where, and we actually, essentially, like, we use computers to guess what the exact force is. And it's actually one of the coolest things I did in undergrad is that I actually got given an antibody, and we had a particular thing that had to bind. And the antibody itself was not perfect, and we had to change like amino acids in the right place to try and make the antibody be able to actually bind better to the protein. And essentially, proteins are already about what's called energy minimization, which is making something take the least amount of energy to exist as possible. And
that's kind of like the folding at home, isn't it?
Exactly. That is exactly what that
gamified it.
Yeah, that is exactly what that whole process is about. That it basically folding proteins so that they look like they actually do. Because the problem is that when we do these crystal structures, or if we know about the structure and the basic shape, that doesn't always mean that we have the exact folding correctly, because proteins actually don't just appear. The human body has all of these ways of kind of manipulating and folding them from what they would be exactly if they were the natural. So this is actually a really cool picture here where it kind of goes from the crystal to the X-ray to the sort of the general shape. So the electron density map is the thing I'm talking about when it comes to like the force of the thing. So all shapes are basically created by these forces, because molecules themselves are so small that if molecules would just go right past each other if it wasn't for the fact that these forces exist. So yeah, very deep dive on a lot of stuff there, but
well, I mean, crystal, like elemental crystals are just the macro scale versions of how the atoms connect together.
Yeah, exactly.
So that the atoms might line up and connect in like a lattice structure, and then as they do that, you extrapolate that, millions of times, and then you actually get a crystal.
Exactly.
So a crystal is just a repeating structure.
Yeah.
And so if you have a pure, let's say, gold, or pure gold,
or insulin, if we're talking about proteins, say, because that was the first protein to be sort of fired at, I think.
So any repeating structure, if you repeat it multiple times, eventually you get to macro scale where you actually see it.
Yeah.
And then, so if you held in your hand like crystalline iron or something, you are holding the macro scale of how all those line up.
Exactly. Yeah, it's a really weird thing to think about, because like 99.9% of existence doesn't exist. It's like empty space. And then
if we talk about scale, we don't exist at the smallest scale or the biggest scale.
Yeah.
Like the smallest scale is so incredibly small compared to us, and the biggest scale is so incredibly big compared to us.
Yeah.
That like we exist in this weird middle point, and nothing in the nothing fundamental in the universe is scaled to us.
No.
We just exist in this weird point where we can glimpse both sides.
Yeah. So Purdymouth had this interesting comment: to see protein is to see the face of God. And that is definitely an accurate description of how difficult it is to really like see what something like a protein looks like as basically the smallest kind of building block of life. And I kind of like that analogy of us being sort of on the end of like small and large, because the last story for today is actually about a comet which is called C-2019 Y4 Atlas. And the reason it's called Atlas is it was actually discovered in 2019 from the Asteroid Terrestrial Impact Last Alert System in Hawaii. And so, as you can imagine, this actually is a defense sort of initiative for the Earth. And its reason, obviously, is to get asteroid detection so that we hopefully find asteroids long enough that we can do something about them potentially impacting the Earth. But in this particular case, it discovered a comet. And the interesting thing is that this comet is actually getting brighter all the time, and a lot more brighter than we might expect. And apparently, currently, it is so bright right now that if you had a pair of binoculars, you could actually see it, which is really cool for something that is quite far away. And
which hemisphere, though?
The northern hemisphere, unfortunately.
Ah.
Yes. So basically, at the moment, what is happening is the comet is moving towards the sun. And as this happens, the comet gets increasingly hot. So even though space is cold, being heated by the sun is a lot. And so what happens is these comets essentially burn, and the ice that's on them creates these beautiful tails of debris as the sort of the ice turns into vapor, and some of the dirt on this comet gets captured and makes these beautiful colors. And apparently this particular comet will be a really bright, slightly green color. And its peak brightness is going to be towards the end of May. So I'm guessing that's as it reaches as close to the sun as we can see it before the glare of the sun makes it disappear, and then it moves behind the sun. And I'll actually put a picture up in a second of the
and assuming it survives the trip, we'll see it again when it comes back around.
Well, here's the thing. Actually, we may not see it, potentially. So this thing has a very large orbit. And they actually think that this could be one of the more spectacular comets that we'll see since Hale-Bopp in 1997. And one of my greatest memories of a kid was being able to see that in the night sky. Really, really fascinating. But there's actually an interesting thing about this comet. So it turns out that this comet is following basically an almost identical path to this famous comet from the 1844. And essentially, that means that this comet has a 6,000-year orbit that actually takes it technically out of our solar system. So we won't be seeing it again.
No, what I mean is because it follows an orbit around the sun.
Yes.
So as it comes around back from behind the sun, but we can see it again, assuming it still has ice on its tail, we'll be able to see it probably July or August as it comes back around and heads out into space, into deep space. Yes.
So what I will do, I can put up this thing. So the interesting thing is, as I get the picture for us, is that because this comet is taking the same trajectory as this comet from 1844, what they think is that these fragments, like it's very unlikely that two different comets would follow exactly the same trajectory, right? The same orbit. And what they think is that basically a massive comet existed at some point and broke apart. And essentially these smaller comets have been over time, and this is, hundreds of years. So imagine the scale involved in this. These smaller comets have been following the same trajectory. And every now and then we might actually see these comets following this particular trajectory that all came from this original massive comet, essentially, which I find really cool.
That is cool.
So let me find for you the.
So as you look for this, I want to point out that historically comets have been seen as portents and omens of disaster and things. And considering where we are in what's happening, the fact that March, we had potential for war back in January, February was fires. March has been coronavirus. April, there's a comet.
Yeah.
People are going to start lining things up the way that people do.
I'm sure they will.
Oh,
So let me put this on the screen here, and it was unfortunate for you, Ben, that you probably won't be able to see it. But the cool thing is that it has these really cool examples of why the planets don't just sort of go in an exact circle, and the sort of, that idea of like retrograde. So the diagram basically shows this period of firstly years in these like little spirals in the sky, as obviously it kind of, because it's moving from so far away and the Earth is doing the rotations, it has a very tight curve in the sky because, of course, it's so far away, And now it's coming into view, and basically for the next 3 or 4 months, basically peaking in May, and then as it moves away again through through like June, July, you'll get another glimpse of it after it goes around the sun. You see this lovely arc through the northern hemisphere, unfortunately for us. And then again, we'll eventually get to see a very faint version as it moves away in a particular direction in these kind of tight spirals, which I think is really fascinating. Like this is a beautiful picture. And it's just like, it's a really cool version of, like the Kepler sort of, the Kepler laws and stuff, and how his laws of motion basically were able to describe why orbits sometimes move backwards in the sky and then back again. So yeah, basically for the past few years, this comet has been doing like a little loop in a very tight part of the sky, and then eventually it will do this nice little sort of circle around roughly where the sun would be. And I think that's really awesome. So if you get a chance and you're in the northern hemisphere, I do believe we can see it from the southern hemisphere, just not quite as well. It's probably going to be low on the sky.
Yeah.
Which means it won't be as apparently bright because we'll have too much pollution in the way. But if
we might also only be able to see it at like sunrise or sunset or something.
Exactly, it just basically pops up at awkward times. But if you get a chance to look at it, all you need is a pair of binoculars. And to be fair, we're all at home at the moment and we have not many things to do. I would recommend everyone to get a good pair of binoculars and just look at the sky sometimes. You'd be surprised what you can see with just like a good pair of binoculars. Obviously not those sort of like cheap ones that you take out walking just to sort of, look, but the ones that are kind of have decent sized lenses. You can really see some cool stuff if you look through them. And, even just looking at the moon or Jupiter or Saturn can be a really satisfying experience with just a pair of binoculars. And obviously they're a lot easier to use and set up than, say, a telescope. So, Well, Ben, thank you so much for joining me today. Obviously, you are the host of the Non-Peer-Reviewed podcast, which I am a frequent guest of and always have an absolute blast. So why don't you tell people how they can find out more about that and what it's all about?
I'd love to, and thank you very much for having me on the show, Ross. It's been good to come back. I was here for the first episode, as you mentioned, and a bit smaller and quieter compared to having you and your other co-host. But yeah, it's good to come back and do a video show. So, as you mentioned, I host a podcast, which is audio only. I host the non-peer-reviewed podcast, non-peer-reviewed podcast. It is a mostly weekly, light-hearted look at science and science stories and questions I find through Reddit and the wonderful / terrible Yahoo Answers service.
Yep.
We have a good time. It's myself and 2 guests. The slogan for the show is the Non-Peer Review Podcast. It's our name and our disclaimer.
Yep.
It's a good time. We have fun. You can find it on all podcasting apps. You can find it on Spotify. You can find it on SoundCloud, the social on Twitter and Facebook. Yeah.
And it is a lot of fun. Like we talk about some news stories kind of similar to what we do here, but we also have these kind of like very like fun segments, where we look at sort of interesting questions that have been asked on the internet and sort of, I always like the Ig Nobels as well when we sort of bring those up when we get a chance. So that's always fun. And it's great. And I really encourage you to check it out.
Next week's episode, we have a researcher who looked at the effect of the ABC show, The War on Waste, how that had an effect on people, if it made changes to their behavior. So I got her to come on the show, Rachel, to come and talk about her research. Yeah, it's really good. That's come out next Tuesday.
Yeah, that's really great. So yeah, people go and check out that. The Science Club show does have a YouTube channel called the Science Club Network, where we post daily, little daily videos just discussing one science topic. We also have a segment called Dose of Science, and the first one of those is called Flattening the Curve. So you should check that out. It's all about, you know, social distancing and why flattening the curve is really important, which we've talked about on the YouTube channel and this show a lot. If you liked the show, give us a follow on Twitch, subscribe on YouTube. You can check us out on Twitter and Facebook in all the usual places. Science Club Show and Science Club Network are the sort of things to search for there. And until next time, 2 weeks' time, we'll be live again. Thank you very much for joining us, and we will see you then.
See you.